بسم الله والحمد لله والصلاة والسلام على رسول الله. اللهم علمنا ما ينفعنا وانفعنا بما علمتنا وزدنا علما وعملا صالحا متقبلا يا رب العالمين وافتح علينا فتوح العارفين. So hello my friends, today we will be learning about the DNA replication. But before we start, I want you to know the basic enzymes that they are included in a DNA replication. So what are these enzymes? Let us learn them together. The first one is called the helicase enzyme, or known as the unzipping enzyme, because it's responsible for breaking the hydrogen bonds between the nitrogenous bases in a DNA. The second enzyme is called the DNA polymerase and is known as the builder, because it's necessary for the building of the new strands of the DNA. The third one is the primase, and it is known as the initializer because it's important for the DNA polymerase to figure from where does it have to start the DNA replication to have a primer. This primer is initiated by the primase enzyme, and it is constituted of a short sequence of RNA bases. The fourth enzyme is the ligase enzyme, and it's known as the gluer because it's responsible on gluing or joining the fragments of a DNA for the DNA replication to be completed. But before we jump for the details of a DNA replication, we have to know where does it occur. So firstly, if we are talking about a eukaryotic cell, it occurs in the nucleus, while if we are talking about a prokaryotic cell, it occurs in the cytoplasm because they do not have a true nucleus. As an additional information for you, in the next figure, it represents the bacterial cell as an example of a prokaryote, while in the first one represents the cell of an animal, for example, is an example of a eukaryotic cell. And we are also concerned about when does it occur. So the cells make it when the two daughter cells have to take a copy of the DNA. And specifically, if we are talking about a eukaryotic cell, it occurs before the meiosis and mitosis in a stage that is called the interphase. So let us take a closer look about the DNA replication and to know the role of each enzyme. So as we have said that the helicase enzyme needs to work by breaking the hydrogen bonds between the nitrogenous bases to form the replication fork. Let us take a look about the legend in here. The red stands for adenine, yellow stands for thymine, green stands for cytosine, and blue stands for guanine. And for the DNA polymerase to work, it has for the DNA original strands to be separated and to kept separated by something that is called the SSB, or single strand binding protein. Okay, and also we need to prevent the supercoiling of a DNA by an enzyme that it is called the topoisomerase enzyme. The supercoiling means the overwinding of the DNA strands, which is something that needs to be controlled in a DNA replication for the helicase enzyme to work. And since the DNA strands, the original strands, are presented in opposite direction, one in 5' 3' and the other in 3' 5', result in the new strands of different properties. One which is the lagging strand and the other which is the leading strand, which we will talk about in details in the next slide. So let us look at the origin of replication. By the way, we have multiple origins of replication in eukaryotes, while only one origin of replication in prokaryotes. And as I have said earlier, we need the primase enzyme, which is necessary in initiating the process, by which it synthesizes the short sequence of the RNA bases, which is called the RNA primer. And it's also necessary for the work of the DNA polymerase to know where does it have to work exactly. And as you are seeing in here, we have the template strands from the original DNA. One starts with a 3', 5' direction, and the other is in a 5', 3' direction. The same is for the new strands. One in a 5', 3' direction, and the other is in a 3', 5' direction. Because the DNA polymerase has an interesting property is that it works only in the 5' 3' direction, we have a leading strand and a lagging strand. Firstly, let's clean all of this. Okay, why this is called the leading strand and this is called the lagging strand? This is called the leading strand because the DNA polymerase directly and continuously synthesizes this new strand in the 5' 3' direction by putting directly the DNA bases, while in the lagging strand, the DNA polymerase has to keep racing up next to where this unwinding happen, which result in the formation of series of small chunks or fragments called Okazaki fragments. Then, after the DNA is replicated, an enzyme that is called exonuclease has to come and to degrade and remove all the primers from both strands. Then the ligase enzyme is necessary for gluing. Remember, we called it the gluer. It's necessary for joining these strands of the DNA, new strand of course, to have a continuous strand. So at the end of this replicating process, we will have two double helix DNA molecules from the original DNA, and it is called a semi-conservative DNA because each contains an old strand and a new strand. Old strand and a new strand. That's why it's called a semi-conservative replication of the DNA. An important information we have to know is that the deep research and the deep understanding of the DNA replication has provided a wide array in the medical field for the protection and for the stopping of the DNA replication inside a pathogenic bacteria or inside a human cell cancer. Also, I want you to pay attention to an important property about the DNA polymerase is that it rarely does a mistake, which means it has a proofreading capability. What's meant by that? Let's take this example. The DNA is responsible in synthesizing the protein, and this protein is important in the function of our vital organs in the body. As an example, the intestine, the stomach, the liver, the lungs, the brain, etc. We took this fragment from the DNA, which is the normal DNA, of course, that will yield a normal protein. But in case this DNA polymerase missed a certain nucleotide, for example, what will happen? This mutation will cause an abnormal protein in most cases, and a disease might occur, isn't it? This is an important issue concerning the DNA polymerase. Now we have reached the end of our video, so do not forget to like my video and subscribe to my channel, and of course do not hesitate to state any question you have down below in the comments. I'm willing to answer any of it. Thank you!